for the nice introduction, and also I take the advantage of the moment to thank people that are hosting me in this university and people that I collaborate with. I see lots of them sitting here in the audience. The task for me today was not easy because I knew that the audience would be mixed. That is, I need to have the interest of specialists, so chemists, and also the interest of other people. I hear like a little whistle when I talk. I hope it doesn't annoy you. So, and the other people that don't know probably anything about chemistry, so I hope I can I can get them on board as well. So I've chosen a topic that is a thing of general interest. That is also the topic that I actually normally investigate under the chemical point of view. The topic is neurodegenerative diseases. Today, we have been recently massively affected by the COVID emergency, but you have to know that the population of the world is getting more aged. There are countries where 1/3 of the population is already over 60, like Japan, but even Italy is not really that good under this point of view. So very old people are living in these countries. And of course, there are lots of diseases, neurodegenerative diseases that are associated with age. So these sort of studies have a high impact on society because taking care of people that are not healthy for 20 years or so is a big issue on the economic, but also, of course, health problems. So under any point of view is very difficult. So I hope you understand the importance of this sort of thematic. University of Catania, just very briefly, is situated in Sicily, for those that don't know. I specify for people that are not even European, because it occurred to me lots of times people don't even know where Sicily is. Sicily is in the middle of the Mediterranean Sea. There is a massive volcano there. That's the view you see when you land or you take off from Sicily. That's the harbor. That's Etna. That actually, I was surprised. It was in the news, in the French news yesterday, because we had an eruption. For us, it's normal, but because what was not normal is the bad weather we had. So there was cloud and ash. So they closed the airport because they couldn't see where the ash was. And lots of people got affected. But anyway, fortunately, I was here in the sun, so no problem. This is the main square. It's a Baroque city. So let's go into the details of the talk. I thought to let you know that there is a big effort by the scientific community regarding, of course, medicine. So a way to tackle those diseases is, of course, to find a drug that can somehow heal or at least slow down the neurodegenerative process. Of course, in order to find a drug, there is lots of study involved. Usually, to have a drug, it takes about 20-25 years from the initial chemical study to the actual clinical phase, and then eventually to sell it to people. Because, of course, you need to first to understand the target, so the enzyme or the molecule that you want to target, then understand how this is misregulated in your disease, and then have some molecule that might interact and stop the sort of unhealthy path, if you want. And then, of course, have all the trials, first in cell, first in vitro, that is what I normally do. Then I collaborate with lots of biologists, because then we need to test the drug in cell. And then after in cell, you go in vivo, for example, with mouse, rats, and so on. And eventually, if you pass all these phases, you go at clinical phase with human. That is very often the most difficult phase to go. And with diseases like Alzheimer disease, at the moment, sometimes in the news appear news that they say there is a new drug and so on, but so far there is no drug that is capable to heal Alzheimer disease. So some drugs now are available to slow down a little bit the process or the cognitive deficit, but no drugs are available at the moment to stop the biomolecular mechanisms involved in the development of the disease. So if you get Alzheimer disease, basically you're going to die within 15 years, no matter what, at the moment. As you can see, so medicinal chemistry is involves lots of different branches of science. Here I just mentioned some of them. Of course, there is all the medicinal stuff that is missing. This is things that we try to do in my lab and when we collaborate with the others. As you can see, the reason for involving all these people is because things in vivo are very complicated. So I basically was trained as a chemist to understand chemical reaction, but then when you transfer those knowledge in the even in-cell system, things gets very complicated because in your little vial you usually mix up 2 or 3 things, while in the cell is full of things, and we sort of manage to try to categorize, if you want, the different things. So you have the genome, that is the DNA, contains information on which protein should be synthesized. So you have the transcriptome, sorry, it's difficult for me to say. It is basically those molecules that tell to the cell which information needs to be transferred, and so from DNA to go to protein that gets actually synthesized. Then there is the proteome, that is actually the ensemble proteins that are present, and that's what normally is studied by the technologies, the technical instrument that I use in my research. So I'm mainly focused on the proteome, and then there is also the metabolome, that is all the metabolites that are the small molecules, because proteins are big molecules. Then there is the small molecules that also affect how the protein works. And finally, there is also the metallome. That's another field of research that is very interesting. That is basically the role of metal ions in living organism. You all know that we have lots of metal ions that help the enzymes and the cell to keep the good work going. And the regulation of this metallome has proved to be, in recent years, very important, as well as all the others. So, of course, when you study this sort of system, you need to try to have a bigger glance at the system rather than just focus on your own vial. That's what I tell to myself. That's why I like collaborations with people having different expertise. Regarding the metallome, I put these slides just for the large audience here, because I think this is something that few people think about, and I think it's worth to have a minute of reflection on this. That is, the way we are made of, the molecules, the proteins, and everything that makes life the way we know it, is actually due to the relative abundance of the elements that are present on the Earth. because when life started on Earth, it basically took advantage of what was available. So it's not a case that our molecules are made mainly of carbon, nitrogen, oxygen, hydrogen, and all these sort of elements that are called bulk biological elements. So if you can imagine there is a life somewhere else in the universe, life can be completely different, not just the shape of the organism that live there, but if there is a planet that have different natural abundance of the elements, because the natural abundance of the element is not the same all over the universe, right? It is the same, but for example, in our planet, on the Earth crust, there are specific elements that have a relative abundance very different from the rest of the universe. For example, oxygen. Oxygen is not really, the abundance of the oxygen we have on Earth crust is not the same as the rest of the universe. So every living organism take advantage of the elements that finds available. And that's also a way to look at evolution in life. Before, we had a reducing environment when the Earth started its life. And the organism that developed used this reduced environment. So there was, for example, copper 1 rather than copper 2 and so on. And then once the Earth changed this relative abundance of element because plants started to inhabit the planet, we had more oxygen and so on, the system, the Earth had oxidative environment, oxidative stress environment, that is not a reductant environment anymore. Even the abundance of element and their oxidation state changed. So the way we are made, that was the little thing I wanted to say, is basically due to the relative abundance of the elements. And also even the disease develop depending on how much of a certain metal ions there is. Saying that, I don't know, cobalt is toxic or is good for you, and copper and zinc, it doesn't really make any sense if you don't specify the quantity of the specific metal ions. So for any metal that we have in our body, there is always a range where this is in Italian, actually, I didn't realize. There is also always a range of healthy amount, and then if you have too few or too much of the element, you get a disease. So it's not a question. I'm saying this because in many studies, even for example, Alzheimer disease, because Alzheimer disease is correlated with copper, zinc, iron. You find in the literature there is loads of papers saying there is too much copper there is too few zinc, and so on. Very often it's a question of not just how much there is in there, but you have to be in the good range, and you have to compartmentalize. I cannot speak today. Basically, to have a separate amount of copper in certain compartment and certain amount of zinc in other compartments. So it's all a question of having the right amount at the right place, rather than just taking the post-mortem brain of a diseased patient and analyze the full amount of it. So now let's focus a little bit more on what I do and what I have been studied. I've been interested, as I said before, in proteins. And proteins generally have this scheme I found that suit what I want to say. That is, basically you have the synthesis of protein, but also the protein needs to be properly folded for it to function properly. And this folding mechanism, of course, can fail when the protein is damaged or aged. So you get a protein that is misfolded. When I say folded and misfolded, for those that don't know, it means it is the quaternary structure of the protein. So besides its linear structure, there is also a shape that the protein must have, the conformational state. And it can be lost if there is a disease. But we have lots of repairing tool that degrades the misfolded protein. So you can have an healthy, unstressed situation where you have the synthesis and the degradation of the aged protein. You can have an healthy, stressed situation where you increase the stress of the protein due to disease or something else, but then you have the body and the cell react to this stressed situation and adjust so that you still don't get a diseased person, because your stress response increases, and so you cope with the abundance of misfolded protein. But then you have a situation where you get stressed because of age or disease, and the body cannot cope anymore to get rid of misfolded protein. So this is a general mechanism. I'm not talking about which protein and so on. I'm just saying that's why the title of the talk, holistic approach. This general approach to neurodegenerative diseases is because if you change the protein of interest, you basically change the disease. If we talk about misfolded amyloid beta peptides, we will have Alzheimer disease. If we talk about misfolded alpha-synuclein, we will have Parkinson's disease. Even misfolded insulin, although insulin is a bit different, but still you find that the insulin aggregates and you get insulin aggregates in diabetic patients. So it's a general mechanism, and when you change the protein, you change the disease. So what are the intracellular pathways that normally are used by the cell to remove this misfolded protein? They are mainly two. One is autophagy, and the other one is the ubiquitin-proteasome system. Both mechanisms are widely studied because you understand that if you can somehow increase the way the cell degrades misfolded protein, you can do some good on cells that are, how to say, that are hindered by the misfolded protein around. So basically, regarding the ubiquitin-proteasome system, the importance of what I'm saying is demonstrated by the fact that these guys got the Nobel Prize because they explained how it works. And also the Nobel Prize for autophagy. So they are the two principal mechanisms that people nowadays are studying, because if you can increase the activity of this degradation, if you increase the degradation of the misfolded protein, you basically ameliorate the situation, and you can think about curing diseases. So how the ubiquitin-proteasome system works? Very briefly, once you have the misfolded protein, it's called the ubiquitin-proteasome system because ubiquitin is a small protein and is used by the cell to tag any misfolded protein. There is a system of the enzyme here that help to getting this covalent linkage on the misfolded protein with ubiquitin. So we will have the misfolded protein now is tagged with a polyubiquitin chain. And once you tag the protein, then this protein is recognized by the proteasome. This is a big molecular entity made up of several proteases that are enzymes capable to fragment misfolded protein. So the the tagged protein, and only the one that are tagged, gets into the proteasome. So there is a regulation mechanism in the open enclosure of this entrance into the proteasome. And then they get released into oligopeptides that are not toxic anymore, and the cell can eliminate them easily. The proteasome is made up by 3 different units. I mean, there are lots of studies on this, so I'm not going into the detail of all this. But our contribution, for example, from my lab by mass spectrometry was, well, first we studied, this was in collaboration with Rett. They are biochemists, so people who do native gene electrophoresis, that is nothing else than seeing the way the proteasome is done in its native form. So it's not because normally when you do gene electrophoresis, you need to destroy the native form of the protein by using information. So here they did this native gene electrophoresis to see which particle of which particle is made of the proteasome, basically. And they did this, but they didn't, in gene electrophoresis, you just get to this sort of bands where you see the weight of the different particles. Mass spectrometry is good. I mean, you can't read very much of this, but basically it tells you which proteins are present in certain samples, let's say. So what we did was analyze the proteasome of healthy people, special cells that they had that collected, and also the proteasome of people affected by a disease that is called Rett syndrome. Is a genetic disease, actually, to see if there were difference in because these patients accumulate a certain protein. So we thought that could be a misregulation of the proteasome, and that's what we found. I mean, the thing we read are all the proteins that are different in terms of abundance, but also some of them were completely missing or were changed respect to the healthy subject. And so basically we could correlate the disease. So this is not just a way to understand the mechanism of the disease, but also to diagnose the disease. So if you get material, I mean, I know it's not a simple analysis, but by looking at the proteasome units of the patients and of you can distinguish between patients that have the Rett syndrome and healthy subject. So a way to tackle the misfunctioning of the proteasome is to find molecules that can, as I said before, increase its activity. And there are some already in that are sold, both for inhibit the proteasome and activate the proteasome. This is weird to explain because depending on the disease, you might want to activate the proteasome or inhibit it. If you get cancer, for example, cancerous cells are very prolific. They develop themselves very quickly. So if you want to stop them to proliferate, you inhibit the proteasome so that the proteasome inhibited cause accumulation of the misfolded protein in cancer cell, and the cell die. There is a way to, of course, to make that the drug goes, especially in the cancerous cell and not in the healthy cell. So they sell already drugs that inhibit the proteasome, and now the new prospect is to have drugs that activate the proteasome for maladies such as neurodegenerative disease, as I said before. And because the phase clinical trial is very long, there is a lot of studies that want to repurpose drug. that is basically drugs that are already used for some diseases. They are proposed for other diseases because the way they act can be bivalent, for example. And one case of this was there are lots of drugs that are nowadays tested to see if they can activate the proteasome based on, of course, on molecular structure. So people look at molecular structures that can fit, especially the open close gate at the entrance of the proteasome, so that they can stop or fix the proteasome open and close conformation to facilitate the entrance of molecules and facilitate the degradation. So basically on the structure, we also tested some molecules already used in other diseases to see if they could activate the proteasome. This is a screening we did first in vitro. You can see the structure of this, but these are all sort of organic, small organic molecules. Some of them slightly mutated, some of them, particularly those three, activated the proteasome. So after this test, we selected some of them for activation. The most promising was aminopyrin here, because it activated by at least 30% the proteasome. We therefore selected some of them. The problem, of course, I'm going quickly now. The problem is that some of them were not even soluble in water, so it was difficult to study them in cell, because that's what the chemists encounter when perform a study. Sometimes there are problems involved with the solubility, for example, so you need to change solvent, but then solvent different from water are very often toxic to the cell and so on. So we selected four that were basically soluble in water, and among those, by the testing that we did, the aminopyrin was the most promising. Aminopyrin was promising to activate the proteasome. So then we tested in cell if when you do amyloid beta peptides, usually the cell gets stressed and eventually dies, because accumulation of amyloid beta peptides is toxic. So we added amyloid beta peptides to cell, and of course you register, you see a loss of cell viability, so lots of cell died. Of course, then, if you go with aminopyrin, that is this bar here, the cell restore, well, if you do, of course, another parallel experiment, the cell live better. So aminopyrin seems to protect the cell from amyloid beta toxicity. These are other compounds that we tried, and also we utilized this bortezomib, that is a strong inhibitor of the proteasome. So if you put aminopyrin, amyloid beta, and bortezomib, the cells still die. That means that the mechanism of action of aminopyrin involves the proteasome. So this is the sort of trials that you do when you have to test those things in cell. And then, of course, by mass spectrometry, we also checked that. So we checked that the proteasome was degrading amyloid beta peptides, and you can follow the degradation of the amyloid beta peptides by mass spectrometry, by looking at the fragments that are generated, and then when you put aminopyrin, you can compare the fragments that are generated, and you can also compare the speed of degradation. This is something I've been working on for many years. That is the monitoring, and I plan to do even here now. That is the monitoring of enzymatic activity by mass spectrometry. I just want to give you a small explanation of why it's good to monitor enzyme activity by mass spectrometry. So I didn't prepare any slides for this. I just want to give you a glimpse of the importance of this. Basically, when you do normally, how do you monitor enzyme activity? What you do is you use a fluorescent substrate, for example, and then you put the enzyme. When the enzyme cleaves the substrate changes fluorescence, and you monitor the enzyme activity by monitoring the fluorescent, the change in fluorescence. By doing this, though, you miss the information about cleavage sites, you miss the information about where the enzyme cut, but also if the cutting of the substrate is changed, not only in terms of quantity, but also in terms of generation of fragments, because some fragments of the main protein can be toxic themselves, can have different biological activity. So monitoring all the fragments that are generating and how this change when you modulate enzyme activity is something very important in science. So that's why we monitor the enzyme activity, in this case, the proteasome activity by mass spec. All I have said, we started to collaborate with Lisa, Olivier, even Juliette is sitting there, because we thought about synthesize some molecules that could be activators of the proteasome. That is this small peptide, it's called TAT1. But also because this other dipeptide, this is called carnosine, was reported to be an activator of another enzyme, it's called the insulin degrading enzyme. We thought about synthesizing this sort of chimera peptides, so containing two different features, both the feature activating the proteasome and the other one activated the other enzyme, the carnosine molecule that activates the insulin degrading enzyme. We published already some work on this, but unfortunately, we didn't have very good result in terms of biological response. So I wanted to put this just to highlight the fact that it's not easy when you go in cell or in vivo to obtain good results of molecules that in principle might function. So we're still carrying on this. And then I know it's a bit late, so I just want to give you a little glimpse on biomolecular interactions method. And I don't know how much time I have, I think about 20 minutes. So I've been, because I was always interested in studying the molecular mechanism involved in Alzheimer disease, because I realized in life, as a scientist, you have to choose between becoming an expert on a technique. That is something many people do. I just come from a conference in mass spectrometry where there are lots of experts in mass spectrometry, and there are people who do only mass spectrometry, so they get samples from other people and they do by their techniques, their experiment. And that's something I like too, but I think the other way could be being an expert, well, not an expert, but at least being focused on a problem rather than on a technique. So if the problem is, for example, in my case, neurodegenerative diseases and the biomolecular mechanisms involved in neurodegenerative disease, of course, you cannot use only just one technique. You have to span your expertise so that to cover other techniques. And there are many techniques that investigate biomolecular interactions that are those things very important for this sort of diseases. Well, I'll just skip this because I realized there is no time. Here. And these are the experimental techniques that you normally use to measure biomolecular interactions. So as you can see here, there is optical and non-optical technique. Among the optical ones, there is SPR, that is surface plasma resonance. So I just want to, because I worked very much on this, I just want to give you a glimpse of this. So how does it work? You have a prism here. So you have a gold surface, and you get some light from behind, and then you register the light that is reflected by the surface. So if it was a total reflection, you would get just the same intensity that you had on the source. Instead, depending on the angle of this of the incident angle, basically, there is a certain angle that you get an absorption of light. This is due to the fact that you are exciting. So now we go in the field of physical chemistry. You are exciting the plasmons of the gold surface that are some collective oscillation of the electrons that are free to move on the gold surface. So basically, this is the physical principle. And then whenever you attach something on the surface, you will observe this minimum that moves. This is because this is the equation that rules the phenomenon, and it depends on the angle, on the actual angle of at which the light is there. So if you change this angle, you change the refractive, the refractance of the light, and whenever you attach something on the surface, you will change this angle. So basically, this is just to tell you that you can monitor whatever happens in the surface by fixing the angle and looking at the different intensity in the light that is reflected from the surface. I mean, might seem complicated, but it's actually quite easy because the user, what does he do? Is you put your detector here and you observe the intensity of the light. So whenever there is association on the surface, or there is a biomolecule accumulating on the surface, you get an increase in the signal. Whenever things detach from the surface, so they go away, you get a decrease. And you can follow this in real time. So you can actually have these curves that are called sensogram for a specific interaction between something that is immobilized on the surface and something else that is flowed to the surface. So this is the technique I've been used for the past 10 or 15 years to study molecular interactions. Of course, why do you want to study molecular interaction? Because whenever you are interested, for example, on the interaction between an enzyme and its substrate and how this can be modulated by a drug or something else, you can study the kinetic parameters involved in this interaction by this technique. And then by comparison, you understand what's going on, basically. So this is the model that you build. You have your molecular part. Here is called A, UB, and this is the complex. So this is an equilibrium. You have the K1, the K-off, and then you basically can get the affinity for that particular molecule and lots of other interesting information. This is what we did, for example, with this enzyme, insulin enzyme. So you first attach the enzyme on the surface, and then you can study the interactions, for example, with insulin. Whenever you put another molecule that affects this interaction. So, for example, the carnosine that I mentioned before affects this interaction very much. So you obtain different curves, and then by doing a modeling of the data, you can obtain important information on some biological parameter like the Higgs coefficient. So this is how we normally use the SPR, the surface plasma resonance, to study biomolecular interaction. Of course, you have to know that protein not only interact with other protein that are immobilized on the surface, but there are lots of aspecific interactions with generally with surfaces. So if you have a protein and you have a gold surface, the protein tends to attach to the surface, no matter what is on the surface, because it has a specific interaction that are mainly electrostatic interaction with the surface that is negative and with the positive charges of your protein. This is a problem with SPR, because if you do a biomolecular interaction between a protein and another protein mobilized, you never know if it's a specific interaction or a specific interaction with your protein. So what you normally do, you get a reference channel where you have the bare gold, and then by comparison, you understand what is the true curves of interaction. 2 years ago, we started to do another approach by SPR. So I don't know, I already talked this in a talk here. So I briefly want to mention this for the chemists, especially because this is something that could be interesting for people that want to collaborate with me. Because now we are able to measure the diffusion coefficient of molecules. What is the diffusion coefficient? The diffusion coefficient is very easily, it tells you is an index of how fast the molecules move in solution. So if you have the molecule in water, this will move around, will diffuse with a certain speed. And the speed is very much dependent on the size of the molecule. So bigger molecule move slowly, is like in real life really. And small molecules move very quickly. So by measuring the diffusion coefficient, you can easily understand the size of one molecule. That's at first glance. But really, it's not just the size, it's the hydrodynamic radius. So a molecule can weight, for example, 1,000 Dalton, but even two molecules that weight both 1,000 Dalton can move differently in solution because of their conformation. That's, we go back to the initial message. Because the conformation, if you have a protein that is large enough, can be sort of a globular form, so it will have a compact form, so it will have a small hydrodynamic radius. But if you have a molecule, I think I have, I thought I had the picture of the molecule. No, I don't have it. So if you have a molecule that is elongated form, vice versa, it will have a high hydrodynamic radius. So basically, by measuring the diffusion coefficient, you can you have a direct measure of is Einstein that gave us this interesting formula. You have a direct measurement of the hydrodynamic radius of the molecule, and therefore of the aggregation state and or the conformational state of your molecule. So this is very important because you can test the conformational state or the aggregation state of the molecule by measuring the diffusion coefficient. The way we do it is here, actually, I didn't see. So there is the alpha helix. This is the same molecule in alpha helix structure on the beta sheet, and as you can see, the hydrodynamic radius is very much changed. It's different for the same molecule in two different conformational states. So you can study not only if the molecules aggregate, so you have big entities there because there is lots of oligomers or fibers, but you can also study the conformational state of the molecule. The way it works, I'm not sure I want to go into the detail because it's a bit complicated. I'll try to make you understanding how this works. In a normal SPR experiment, so this is buffer, this is your analyte, and this is buffer again. If there is no interaction between what is flowing in the flow channel and what is immobilized on the surface, if there is no interaction whatsoever, your SPR signal will be like this. And the reason is because, of course, there is a change in the refractive index when the molecule flows above the surface. So you have an SPR response. But as you can see, there is not a sensogram. There is not the curve that goes like that, because it's a steep increase and a steep decrease. There is no interaction. So there is no association phase. There is no dissociation phase. The molecule goes, is seen by the surface while it's there, but then when it goes away, you get this steep sort of a rectangular shape. So this is for something that is not interacting at all. This is what you expect. However, when it goes, when it flows inside the channel, it changes its shape, because the pumping system is so that in the middle of the channel there is a high pressure, and then in the side of the channel there is a small pressure. So the molecules that happen to be very close to the channel wall will go slowly, while the molecules that are in the middle of the channel wall will go quickly. So instead of getting, when they get to the detector, to the actual cell of detection, the detection first see this little part, and then once the thing goes on, you start to see more and more because there is this shape. So instead of getting the curve that I showed you as a rectangle, what you get is this, is a bell-shaped curve. This contains, nevertheless, the information of the diffusion coefficient. Why? Because molecules that, and I have here the simulation. So this is the file. I have here the, well, this simulation. If the molecule moves quicker, it can, while it goes this way, it also quickly moves perpendicular to the flow because it diffuses quickly. So the shape is less pronounced, you see? So we start off from here. Here they are quicker, so it gets here. However, because it diffuses perpendicular to the flow, the shape is less pronounced. If the molecule goes very slow, so this is a diffusion coefficient of 10 to the minus 12. This is, I can't really read the number. I think 10 to the minus 11 and 10 to the minus 8. But anyway, this one goes lower. So if it goes lower, then it diffuses only a little bit perpendicular to the flow cell. So when it gets there after the 40 seconds, it's very pronounced. You see the difference between this one and that one. The one that goes fast is a bit counteractive. It took me a while to understand why I was getting the response I was getting from the machine. Because at the beginning I thought if we diffuse quicker, we should get to the detector with this shape, because it is not, because you have to think that the diffusion occurs in this direction, perpendicular to the flow cell. So the shape of the curve, we're talking about no interaction with the surface. The shape of the curve depends very much on the diffusion coefficient. I have the luck of having a very good physical chemist, mathematician guy in my lab. Well, he's a professor that is probably going to invite if we can next year. And he made all the simulation, all the calculation for me. So we could fit all the experimental data that I knew contained some important value about the diffusion coefficient, but honestly, I didn't know how to fit the data. But he did the script, he fit the data, and we could calculate the diffusion coefficient. Amazingly, it was always the same as the one reported in the literature for the molecules that we tested. So we tested deuterated water, ATP, we tested so these are the calculations, ethanol, so all small molecules. Why small molecules? Because we had this problem of molecules attaching to the surface. That you want to avoid this for this sort of measurement. You want a surface that is as inert as possible. So for small molecule it's easy. For bigger molecule it can be a bit more difficult. But now we worked out how to do it with proteins, so we are publishing this also about proteins. So these are the publication we did already with this new method. We did the carnosine. And also we could see, for example, with carnosine it was interesting because if you do carnosine alone, you get a certain diffusion coefficient. If you put carnosine with zinc, that is very often the way you find it in vivo, the carnosine with zinc form polychains, and these polychains, of course, are much bigger than the carnosine itself, so the diffusion coefficient was much smaller. So this is consistent on what you actually know it should be. And also, if you destroy the carnosine by putting the pH very low or 100 degrees, so you degrade the carnosine in two pieces, the diffusion coefficient goes down. So these are some good results that we obtained and published already. Well, this is what we did with the proteins. Now we made the surface inert, so that you get only diffusion without attaching the protein on your surface, so we can study now even protein. So the take-home message here is confirmation of proteins is important. You can study by mass spec, at least this is what I did in the past few years. You can study by mass spec both conformation and the kind of proteins you have in solution. And now I can study, if you are interested in specific proteins, I can study protein diffusion coefficient at different conditions. So, for example, a protein that tends to aggregate or tends to change its conformational state, I can do those measurements and check the different conditions, what is its diffusion coefficient, and correlate it with the shape and the oligomeric state. So I hope I didn't bore you too much. I tried to be both punctual in something, but also general in terms of message. It's not easy when we have a mixed audience, to be honest, because I didn't know where to stop in one way or the other. So I think it's okay, because I see people more and more tired. So thank you very much again for letting me here talk and for having me here, and thank you very much for your attention.